All right, folks, let's get going for our next session. We have a all-star panel here, three outstanding speakers. We're going to hear from each of them for 15 to 18 minutes, and then we'll still have 20 to 25 minutes for Q&A. You have from our website and the agenda, you have full access to their bios because they all have very long and impressive biographies. I'm only going to touch on the highlights right here. And then I'm going to turn it over to the first of the three speakers. What we'll be talking about here are the replication studies, which is something that Dr. McClellan and Dr. Concanto talked about. Dr. Sebastian Schneeweiss, a professor of medicine and epidemiology at the Harvard Medical School and chief of the Division of Pharmacoepidemiology, Department of Medicine at Brigham and Women's Hospital, is going to lead that. He's been overseeing this study that is comparing randomized controlled trials to real-world evidence studies. Then we're going to hear from Dr. Steven Nissen, who is from the Cleveland Clinic. He's chief academic officer of the Heart, Vascular, and Thoracic Institute and holds a chair in cardiovascular medicine there. He's testified in both the Senate and the House of Representatives on the topic of drug safety, and in 2007 was listed as as one of Time Magazine's 100 most influential people in the world among scientists and thinkers. He's going to talk about how the medical community and doctors feel about the calls for real-world evidence and how they compare to randomized controlled trials. Then Dr. Michelle Johnson-Funk will talk. She's going to talk about some limitations of randomized controlled trials. And she is an associate professor in the Department of Epidemiology and director of the Center for Pharmacoepidemiology in the Gillings School of Global Public Health at the University of North Carolina at Chapel Hill. As I said, it's 10:45 right now. We have these three speakers until 12. We'll hear from each of them, and then we'll have time for Q&A. If you have, when Dr. Schneeweiss is talking, and if you have a question for him, I might be able to work one or two questions in between the speakers, but generally we'll hear from the three and then have a good panel discussion. So thanks to and welcome to all three of you, and I'll turn it over to Dr. Schneeweiss.
Great. Well, thank you very much, Chris, for having me. And as you said already, I'm going to summarize the RCT DUPLICATE initiative, a FDA-funded project, a demonstration project in order to understand when and how real-world evidence can come to causal conclusions by emulating randomized controlled trials as a proxy. This is funded by FDA. Here are my conflicts. Please just pause the recording and once you want to read this. The extensive team that was involved in this RCT replication project at Harvard, at Edion, our colleagues from FDA provided very valuable information, and of course our expert advisory panel. The responsibility is, of course, entirely with the principal investigators. So why do we want to emulate randomized controlled trials with real-world evidence study? This is because we need to demonstrate that we can come to causal conclusions. The big elephant in the room about real-world evidence, of course, is that we might be observing spurious correlations of the disease with the health outcome of interest rather than studying the effect of the medication that we're really interested in. So how can we learn when and how real-world evidence comes to causal conclusions? We need to first identify or establish what is the true causal treatment effect. And if we would know that, if we would know the truth of how well a medication works, then we can do a real-world evidence study and just compare the findings against each other. And if there is a discrepancy, we know there is a problem, and then we can dig deeper and identify what exactly the problem is. Now, how do you establish the truth? What comes closest to that, we think, is well-conducted randomized controlled trials. The challenge then is that is a single randomized controlled trial sufficient to establish a causal relationship? There is variability amongst RCTs. Should we rather rely on multiple RCTs? For the purpose of this project, we identified one RCT each and tried to emulate that one RCT. We also need to assume that we can perfectly emulate all aspects of the RCT. so that we can really disentangle the design aspects from the residual biases. We want to understand the biases and minimize biases, but emulate the trial for this calibration exercise as closely as possible. We don't expect this is every real-world evidence trial replication. That would totally defeat the purpose, because real-world evidence is complementing of RCTs. The difficulties, or sometimes the failures to emulate the RCTs for this calibration exercise may explain the observed differences in the treatment effect estimates that we see between randomized controlled trial and real-world evidence study. And that makes it sometimes very difficult to interpret the findings of these types of calibration exercises. And I should also point out, this is of course not the first exercise, but it's a very specific, very focused exercise. I'm sure in the previous lectures you talked already about the parallel group randomized controlled trial, the workhorse in establishing treatment effects. In medicine, you have a washout period, you have randomization, and people are randomly allocated into a treatment group, an exposed group, and a comparator, and often a regulatory space in a placebo group. And the cohort study is mimicking just that design. You might require that people have not used certain medication in the past. They self-select themselves together with the physician, obviously, into an exposed group and into a comparator group. And this is what we call the new user active comparator cohort study design, which mimics very well this parallel group randomized controlled trial. The trials that I'm going to report on, they all very much look like the scheme that you see at the top of this slide. Very briefly, how do we do real-world evidence study with these with insurance claims data? We have transactional databases. This big arrow is a transactional database that grows with time from the left to the right as time is moving forward. Individual patients contribute as they enroll in insurance company, and then they disenroll from insurance company. And these databases are just growing by the second. More and more information is entered. The information is entered today, but it might contain claims that leads back to services that were provided a year ago in the past. Therefore, these transactional databases are inherently dynamic. On the right end, so the first step is what we do as research. We stabilize the database. We take a data cut out of this dynamic database, and then we look at the individual longitudinal trajectories of healthcare encounters and the affiliated information on diagnoses. If you have hospitals stay here with this patient, it comes a bunch of diagnoses that are recorded with certain coding schemes. The prescription drug dispensing, there again is a barcode reader, and it's getting electronically submitted very detailed and granular information of what kind of medication was picked up at the pharmacy. Then you have the physician visit with diagnostic information. And within that data stream, now we can embed a cohort study where we identify the cohort entry point, often the new use of a drug that is of interest. We follow people over time, and we look backwards in order to establish the health state of that patient. This is all written down. The RCT duplicate project, the methodology details are widely published, and I'm happy to share those slides. So first we started out with identifying these 30 RCTs that were conducted in order to request approval for medical products with medications, actually, with FDA, and a whole bunch. We tried to stratify the sampling a little bit. We want to have primary approval. We wanted to have randomized trials that were designed for supplemental indications. We wanted to identify trials that were designed to go to FDA, but the molecule did not perform as expected, and we call them negative trials here. We wanted to have some of those, and then FDA suggested several trials that they were interested in replicating. We also designed, together with our colleagues from the FDA, a process of how to do real-world evidence study, how to check data quality issues before moving forward in designing a statistical analysis plan, doing feasibility analysis about power and the balance that we achieve between treatment groups in the absence of baseline randomization. And at this point, we could still decide that no, it doesn't make sense to move forward. It is unlikely that this real-world evidence study that we are planning will come to valid causal conclusions. Only then do we register the protocol, we hit the execute button, we run the analysis, share the results with the FDA, and allow the FDA to reanalyze the data, to do sensitivity analysis by sharing the platform that is connected to the data, and they can change definitions like the outcome definition, like the confounding adjustment. And that mimics, again, the process that is happening with randomized controlled trials that are submitting patient-level data of the randomized controlled trial. We're using a standardized software platform that is providing guardrails and provides scalability, transparency, and audit trails so that everything is exactly recorded, what we do with these very raw longitudinal healthcare data. Transparency is written big for this project here. You see these different items that are getting worked on. And you see this feasibility analysis at point 7, 9, and 10. And you see point number 12 is the registration of the study protocol. And only at point number 13 do we do the comparative analysis. So diving right in, we have the first 10 trials. The first 8 of those are second-line antidiabetic medications. The dark red Carolina trial was actually a prediction of an ongoing phase 4 randomized trial, while all the others are replications of trials that were completed at the point in time when we started engaging. You then have antiplatelet trials, Triton and Plato. You have in the second batch of 20 randomized controlled trials, you have the direct oral anticoagulants, or DOACs, that are used in patients with atrial fibrillation in order to prevent stroke. Aristotle, Rely, Rocket AF, and you have the same medications, DOACs, that are used for the treatment of deep vein thrombosis, DVT, Einstein, Recover and Amplify, and then we have heart failure here. We have antihypertensive medications, Transcend and On-Target trials, and we have osteoporosis. You see, we wanted to cut across therapeutic areas, but it is dominated by cardiovascular, cardiometabolic conditions, and certainly cardiovascular endpoints because of the measurement issue. And I should mention here very clearly that we self-imposed, that we limit ourselves to claims data. We did not consider for this part of the project electronic health record data that might have more granularity, but come with another bag of issues that we did not address in this project here. The first observation that we had is in a variable that we observe very well in real-world evidence, which is age. It is almost perfectly recorded in these claims databases. The age distribution is different in the trials compared to the real-world evidence. And keep in mind, we were imposing exactly the same, or as much as we could, the same inclusion-exclusion criteria. We qualified the same age range of participants. Nevertheless, in most of these trial emulation, in the real-world evidence, in the real world, patients are older on average than in the randomized controlled trial. We also observed that it is well known that women are underrepresented in randomized controlled trials. And of course, we see in the real-world evidence studies a higher proportion of women being part of the study. So you see already the tension with, yes, we follow the protocol in spirit, not only spirit, also in the definitions. Nevertheless, we are not perfectly emulating what really happened in the trial. We have a different sex distribution. And that is a consequence of this. But the purpose of this study was to prospectively pretend instead of doing a randomized trial, we are doing a real-world evidence study. So we did not engage in a post hoc reweighting exercise, which is what we could do in order to make the age distribution more similar. And maybe we're going to do this in the future. Here's an example of a Kaplan plot where you see time moving from the left to the right since the initiation of treatment. You see the cardiovascular endpoint incidence on the y-axis. This is the leader randomized controlled trials comparing liraglutide, a second-line antidiabetic medication, against placebo. That is the placebo line, is the black line on top with the highest incidence of MACE, 3-point MACE. In liraglutide is the blue line here. And the difference between those two lines make for a hazard ratio of, what is it, 0.8 or something like that, right? So 20% reduction in the risk for MACE. in the randomized controlled trial. What is important to observe here is that the incidence rates in the real-world evidence study across the board are lower. These two lines from the real-world evidence study are below the two lines of the RCT. Nevertheless, the relative difference is actually quite similar in this example of the emulation of the leader trial, so that the difference between liraglutide, the drug of interest, and DPP-4 inhibitors, which we used in lieu of placebo, because DPP-4 inhibitors have been shown over and over again, they have no effect on MACE endpoints, cardiovascular endpoints, and therefore serve the purpose of an active comparator that is inert with regard to the interest, the outcome of interest. So overall, you get a picture like this, where you have this is the first 20, the next 10 are not quite released yet. The blue dots should ideally align exactly on the green diagonal here, which is the unity point between the results from the randomized trial emulation, the log hazard ratio, versus the finding of the randomized trial. And that, we are getting very close for many of those replications, but some we are further out there, and that, of course, is the important learning experience here. So we have these tables here where you see kind of the first 4 trials: Leader, Declare, EMPA-REG, Canvas, where we come up with a qualitative assessment of the emulation success. We feel we are moderately good in the comparator emulation because we don't have placebo. We emulate the placebo with a drug like the DPP-4 inhibitors. We feel good about the endpoint emulation. We capture heart attacks in similar events quite well in claims data. Now here I have the hazard ratio, this 13% reduction in the MACE endpoint in the randomized controlled trial, and we see this 18% reduction in the real-world evidence with the 95% confidence interval. So great agreement in Leader, as I showed you already in the Kaplan plot. Declare, we have some differences here. It's qualitatively from the regulatory perspective, it is the same. It is superior. The dapagliflozin, which is the drug of interest here, is superior to our placebo emulation. Nevertheless, the estimate, the treatment effect seems to be much stronger in the real-world evidence study than in a randomized controlled trial. And that makes us nervous, and we have to find out why that is. Beyond that, it's actually quite similar. Again, keep in mind, we assume that the randomized control trial is the truth here, and we all know that if you do 2 randomized trials on the same topic area, there might be some variability. The example, the Carolina trial, again, was a prediction. There's a comparison between linagliptin and glimepiride, 2 second-line antidiabetic medications, where we predicted that there is no difference with regard to 3-point MACE, 0.91 with these 95s, but we predicted there's a benefit of linagliptin in reducing the risk for hypoglycemic events. And when the trial was released 6-7 months later at the ADA meeting in San Francisco, where our predictions were confirmed in that there's no difference in the cardiac events, but there is a substantial benefit with regard to hypoglycemic events. The measurement, the emulation of this endpoint, hypoglycemia, is actually quite different between the two. So therefore, it's no surprise that the effect sizes are different, but qualitatively, and for any clinical interpretation, they're clearly moving in the same direction. The anticoagulants, the oral anticoagulants, Aristotle, Relay, Rocket AF, again, overall, actually very nice emulation, very nice success. Then in the point estimate, you see the emulation success be categorized as good for all of them. Now, here is a more interesting case where things didn't go so well. Think of Plato comparing ticagrelor against clopidogrel. These are blood thinning agents, antiplatelet agents, where the randomized control trial postulated an almost 20% reduction in the risk of major cardiovascular events, while we saw only this 8% reduction, and you see the 95 is overlapping the 1. for what it's worth. And when you dig deeper, and I'm sure Steve is very much familiar with this, I was not when we started out, unfortunately. We learned this later, postdoc, that the Plato trial, actually the results were very much driven by the European sites, where there was an overwhelming effectiveness, superiority of ticagrelor over clopidogrel, but not so in the United States. In the United States, it was a failure, so to speak. There was no difference. And these smart investigators dug deeper and found that the concomitant aspirin use was on average in a much higher dose in the United States, and there's some speculation about an interaction. This goes to show the challenges in these trial emulations where you have these multinational randomized trials, but we are working with US data, capturing only a subset of population. So if there's heterogeneity in the treatment effect across nations, across healthcare delivery systems, what have you, then that makes the emulation difficult. Another example is the Horizon trial, that is zoledronic acid against placebo for the treatment of osteoporosis. The outcome is hip fracture. The zoledronic acid is used once, it's an injection once a year. When we looked at our data, the real world evidence data, and that is not surprising, the adherence is much less optimal than we as physicians would hope, and certainly what we see in randomized controlled trials, where you encourage the adherence to the assigned treatment. So we saw almost nobody coming back for a second shot of zoledronic acid after a year. So we decided, well, let's end follow-up after a year. Let's give a little bit of slack. Let's do 1.5 years of follow-up and let's truncate there. All right, that's what you've done. The findings are quite different here. A 40% reduction in hip fracture risk in the trial versus we found only 25%. Again, for regulatory perspectives, it's the same finding, it's superior, but we are certainly curious what's going on here. So if you look at the Kaplan plot, the left lower here, you see the randomized trial and the Kaplan-Meier. What you see, if you truncate the follow-up time at 18 months, you pretend you have only 18 months of follow-up, which is what we have in our real-world emulation, as I just illustrated to you. Then actually, in the first 18 months, the effect size is extremely similar. It's actually 0.75 in a trial, and 0.75 is what we observed. So if we limit ourselves to the period that we emulate, we have perfect success in our randomized trial emulation. The treatment effect of zoledronic acid in this trial it changed over time. The treatment effect got stronger the longer people used zoledronic acid. And therefore, in the 36 months of the trial, we see this 0.59, but we don't have that information in real-world evidence for the reasons that I gave you. But when we look at the 18 months only, I would call this a calibration success. So what we would do, and this looks like a petty post hoc, a cosmetics, therefore I don't want to highlight this too much, but the theoretical exercise would now be actually for the calibration for the first 18 months. We're actually doing quite well, and the point would actually move towards the green line. We do not propose that because we said we are strictly prospective, everything is predefined. So, but I wanted to illustrate that. In conclusion, in this demonstration project where we want to understand, can we come to causal conclusions? If the RCT design could be emulated well, then if the data are fit for purpose, if the proper design analytic options are chosen, then the non-randomized real-world evidence study actually can come to the same conclusion. Most certainly in a qualitative way, but in most cases also in a quantitative way, we're actually quite close. So it is very encouraging news. And of course, these initial findings of RCT duplicate, I showed you just the first 20 out of the 30, indicate circumstances when real-world evidence may offer causal insights when RCT data are either not available or cannot be quickly and feasibly generated. Think COVID, where we always kind of need a combination of RCT and real-world evidence. This is my last slide, and thinking beyond the demonstration project. Well, we have only 30 RCTs. It's still the largest of these types of exercises. Still, it's only 30 RCTs. What about number 31? If we would do 100, what about 101? The way I see this being productive is if we establish a repository of well-documented study that illustrate the agreement or disagreement between RCTs and real-world evidence in specific settings with specific data sources. And that these can be anchor points in the sea of hypotheses, and we can build on that. And if we design studies, or if we evaluate studies that get brought to us in order to make an analogy conclusion that, it's likely that this will fly, or it is less likely that this will come to causal conclusions. It's a bit like case law in the UK and in the US, of course, where you build on prior experience, and you step by step you build your house in your learning. So we have great hopes here. We find these findings very encouraging. It's a great learning experience for everybody, and I hand back to you, Chris. Thank you.
All right. Thank you very much, Dr. Schneeweiss. Yeah, if you could drop your slideshow, and we will turn it over to Dr. Steven Nissen. He is, as I said, from the Cleveland Clinic, and the title of his talk is Real World Evidence, a Dangerous Delusion. So he's going to come at it from a different perspective. Dr. Nissen, are you there?
Yes, can you hear me?
Yep, we can. And
okay.
So looking forward to hearing from you, and I'll turn it over to you right now.
All right. Well, first of all, let me just say that this is a deliberately provocative talk. I'm going to argue this case the way a lawyer would argue a case in court, but it is a cautionary note. And I did title it Real World Evidence, a Dangerous Delusion. These are my titles. And let me first begin by the statement that the term real world evidence, in my view, is an oxymoron, that real world studies should not be viewed as conclusive evidence, certainly not under the current circumstances. I could talk with you for 2 hours about all the things that make claims data unreliable as a source of evidence. So I think a better term is real world data. It's data, it is not evidence. This is a view held by many people, Rory Collins and Richard Peto, two rather well-known, and Sir Richard Peto, legendary statistician, wrote this piece in the New England Journal of Medicine entitled The Magic of Randomization versus the Myth of Real World Evidence. And what they say here is that non-randomized observational studies may be able to detect large treatment effects. However, the potential biases can be appreciable, so such studies cannot be trusted when the benefits or harms of treatment are actually null or only moderate. I wrote this piece in the European Heart Journal entitled, Can We Trust Observational Data for Clinical Decision-Making? And what I said is that big data is often bad data, that these are often eye-catching studies garnering huge press attention. And some journalists and the public are seduced by the large size of the trials of the studies, assuming that the massive sizes of these studies will automatically ensure that the findings are accurate, and they do not. So a clinical trial is a controlled experiment. This is what a randomized clinical trial is. And the whole point is to minimize variability and bias so that you can isolate the treatment as the variable of interest. That is why randomized trials are the gold standard. Now, they have many strengths, and you can read these slides offline, but they minimize selection bias and they minimize confounding, which I'll talk about later, and they allow blinding of patients and physicians, and they are uniformly accepted by regulatory authorities and payers. The problem is that they're logistically challenging, they can be expensive, and they can be time-consuming. Because of these high costs and time-consuming nature of these trials, investigators have sought to conduct simpler and less expensive studies. And in many cases, the results have been disastrous. And I'm going to focus on some of those disasters. Now, I want to talk about hormone replacement therapy, and nobody who does observational trials likes me to talk about this, but it is what happened. is that it was believed for many years that hormone replacement therapy reduced coronary disease. And there were lots of reasons why that might be the case. There were over 30 observational studies suggesting a 40 to 50% reduction in coronary heart disease risk, a massive reduction in risk. And here's one of them, the Nurses' Health Study, 60,000 nurses, 400,000 patient years. And if you look here at the odds ratios, the relative risks, this is about a 50% reduction, in this case, a 60% reduction, published in one of our most prestigious journals, the New England Journal of Medicine, 16 years of follow-up, lots of data. So 1/3 of postmenopausal women used HRT during this era. Second most prescribed drug, 46 million prescriptions. And this was so thought to be so beneficial that a randomized controlled trial was designed to prove the benefit so the remaining doctors who did not prescribe would prescribe. And a lot of authorities fought us when we wanted to do a randomized controlled trial because they argued it was unethical since the therapy was obviously beneficial. The trial was done first in secondary prevention, the Hurst Trial. Postmenopausal women, documented cardiovascular disease, estrogen, progestin versus placebo, double-blinded, 4 years of follow-up, 2,763 patients, and published in JAMA. And what it showed is there wasn't a 40 to 50% benefit. If anything, the trends, although not significant, went in the opposite direction. More events and more mortality compared with placebo. You know, very surprising. There were also many more thromboembolic events, more gallbladder disease, and, you know, it was dismissed. In fact, there was no perceptible impact on prescriptions since hormone replacement therapy was believed to have other benefits. So the needle didn't move when the Hurst trial was published. And so the follow-on trial that Bernadine Healy had the wisdom to push, what she had at the NIH, was the Women's Health Initiative trial. Large trial in women without coronary heart disease. Again, now 16,600 patients followed for 5 years, and it was stopped in 2002 by the Data Safety and Monitoring Board due to excess risk of breast cancer with hormone replacement therapy, also published in JAMA. But let me show you what happened with coronary heart disease. Now remember that the real world evidence suggested a 50% benefit. In fact, coronary heart disease increased, stroke increased, heart disease by 29%, stroke by 41%, pulmonary embolism increased, it more than doubled, and invasive breast cancer increased. These are the Kaplan-Meier curves showing you exactly what happened with the Women's Health Initiative study. This made big news. This is my friend Gina Colada's piece in the New York Times. And she said the announcement yesterday that a hormone replacement regimen taken by 6 million Americans did more harm than good was met with puzzlement and disbelief by women and their doctors. Everybody believed, based upon the real world evidence, that it would go the other way. And the evidence from observational studies was wrong. Why was it wrong? And the problem with observational studies, no matter how you cut it, is confounding. And here's how confounding works. You want to study alcohol consumption effect on coronary heart disease, and that's shown at the top here. But it turns out that if you drink, you're more likely to smoke. And so what happens is that smoking is a confounder. Now you can adjust for confounders, but you can never find all of the confounders. And so people that engage in this behavior of drinking alcohol do a whole bunch of other things that are unhealthy. and looks like they have more coronary heart disease. Alcohol doesn't cause coronary heart disease. It's associated with other behaviors that cause coronary heart disease. And this problem is known as residual confounding. And that's what happened with hormone replacement therapy. Women who choose to use hormone replacement therapy were healthier. They had better dietary and lifestyle patterns that resulted in lower event rates. And despite adjustment for many variables, residual confounding in the observational studies resulted in false positive results. Based on real-world evidence, millions of American women were exposed to a harmful therapy. And the reason I really requested the opportunity to speak to you is that if we follow the current patterns of more use of real-world evidence, we're going to see more disasters like this. It is predictable and it will happen. Real-world studies, in fact, are often a parade of low-quality science. And I'm going to show you a few of my favorite real-world evidence studies widely covered by the media that were misleading. Here's one from the group at the Brigham, nut consumption and risk of pancreatic cancer in women. And what it in fact showed was that women who consumed an ounce of nuts twice a week had a 35% lower risk of pancreatic cancer. Now, if you believe that eating a handful of nuts twice a week can reduce pancreatic cancer by 35%, I have a bridge in Brooklyn I'd like to sell you for a small price. Here's another one. Association of sugar and artificially sweetened soda with kidney function decline, also in women. And if you had 2 servings or more of artificially sweetened soda, you had double the risk of a decline in your kidney function. Nonsense. But this was published in a good journal and got a lot of media attention. And that's the problem, is that you can't separate these poor quality studies from the good ones. That's what happens with the media with these kinds of studies. So big data, in my view, is often very bad data. Here is another example. 3.6 million patients, 9% of the population of the United Kingdom, studying the relationship of body mass index to mortality. This got a tremendous amount of press attention when it came out because of the findings shown here in the box. Is they saw that in the people with moderate obesity, there was actually lower all-cause mortality. And in fact, the authors concluded that grade 1 obesity overall was not associated with higher mortality, and overweight was associated with significantly lower all-cause mortality. And so the media, I can show you the headlines, said, being overweight's not bad for you. Don't worry about it. Go out and eat to your heart's content. It was wrong. Better quality studies, this happens to be one in JAMA Cardiology, have shown you follow for a long period of time, and there is a graded worsening of outcome, morbidity and mortality, as body weight goes up. The problem is that that earlier study got all kinds of attention and was misleading. So when we talk about evidence-based medicine, it's important to understand the hierarchy. And the hierarchy puts observational studies much lower than meta-analyses of randomized controlled trials, smaller trials, a single randomized controlled trial, and then multiple randomized controlled trials. When you have multiple randomized controlled trials, you can generally be pretty certain. When you have observational real-world evidence, you cannot be certain. The randomized controlled trial is the gold standard for a very good reason. It minimizes bias. While we can't do it for everything, we can do it for a lot of things. We must make choices on what evidence to use for clinical guidelines. And for important questions with mortality or serious morbidity, we need randomized controlled trials. If randomized controlled trials are not possible, we must be cautious and vigilant about treatments only based on theory or observation and association. Observational studies, in fact, they suggest an association, but they do not prove causation. And I have a list here, you can read it offline, of all the false positives where observational data gave the wrong answer. It is a problem that has existed for a very long time. When are observational studies useful? As hypothesis generating studies to inform the design of randomized clinical trials. When RCTs are not feasible or the effect size is so large that a clinical trial is not needed. And here's an example. You can't study smoking and lung cancer with a randomized control trial. You can't assign people to smoke or not smoke in a randomized trial. Fortunately, the relative risk was 9. It was a 9-fold worse risk if you smoke. And so you can do a real world study and you can get the right answer when the effect size is so large. It is reasonable to consider real world data as potentially useful if the relative risk is greater than 2 or less than 0.5. For lower degrees of risk, you really can't make any inference at all. So with that summary, let me just say that there is in fact a solution to this problem. And the solution that I've advocated for many years is not to move away from randomized controlled trials toward real evidence, but to do randomized controlled trials as large and simple trials, to do them at much lower costs. And we have done such trials for costs as little as 20% of the cost of conventional randomized trials. The answer is not real world data. The answer is better, faster, and cheaper randomized controlled trials. Now, look, I know I've been provocative, but I wanted you to hear this perspective, and thank you for your attention. You're muted.
Yes, I am. Sorry. Thank you very much. Thank you very much for your perspective. And during the talk, some of the other speakers put some information into the chat about the hormone replacement therapy and the Women's Health Initiative. So there's some links in there if you want to grab that information. Right now, we're going to turn it over to Dr. Michelle Johnson-Funk at UNC Chapel Hill. She's going to talk about some of the shortcomings of randomized controlled trials, but then when real-world evidence can be helpful and be useful. It is 11:28. We're going to hear from her for 15 or so minutes, and then we'll have time for questions. So if you want to get a question in, put it into the Q&A or raise your hand, and we will turn to you during the Q&A session. So Dr. Johnson-Funk.
Great. Thank you, Chris. Are you hearing me all right?
Yes, we are.
Okay, wonderful. Well, thank you for inviting me. If you want to look at my funding sources, you're welcome to review these later. I don't have any conflicts with regard to the material presented today, except that I am a practicing pharmacoepidemiologist with a career in the use of non-experimental studies. So I wanted to anchor this conversation in sort of thinking about what the goal of sort of the classic randomized trial is, and the context in which those are designed, particularly when there's a regulatory decision at play. RCTs need to be designed to answer a specific question, as all research should be. And I would posit that this question is something like, does this specific intervention reduce the risk of a particular health outcome when used as indicated in a particular patient population. And in the context of providing that evidence as part of a regulatory submission, the patients in those studies are selected intentionally in order to maximize the possibility of detecting that effect, such that the background risk of the outcome of interest is maximized, that the adherence to this protocol is also maximized, and that patients are as likely as possible to complete the entirety of the follow-up. Also, other health events that could prevent us observing the main outcome of interest are minimized. And then, of course, patients are excluded in whom unknown risks of side effects would be unacceptable, such as pediatric patients, pregnant women, and nursing women. Obviously, the main advantage of randomization is that it's going to balance both measured and unmeasured risk factors for the outcome, at least on average, if we randomize a large number of individuals. So now I'm going to go on to highlighting some of the shortcomings that come along with being able to conduct a randomized trial. And let me just sort of give the caveat up front that any of these individual shortcomings can be addressed by altering the design, but overall, these are some of the limitations that come along with the classic randomized trial framework. I know that Mark identified some of these early, so I'll go through this relatively quickly, that they are often too small to evaluate the effects on rare outcomes. They are too simple to study interactions and treatments that are changing over time. They're too selected to be generalizable to all patients who will eventually receive the treatment. And I think we saw that in some of Sebastian's slides where he identified significant differences in the age and gender distribution, even of populations, after restricting inclusion criteria to be identical to a trial. They're too specific to assess all of the relevant health outcomes that would be of interest, too short to study long-term effects. I've added, classically, there are only 5 of these shortcomings, but I've added 4 more. So I think they're too stale often to provide relevant evidence that compare a treatment to contemporary alternatives. They're too spendy to assess many questions that are important to public health. They are too slow to identify potentially effective treatments in a timely manner, and they're sample size hungry. So when there are very rare conditions, it can be nearly impossible to enroll sufficient numbers of individuals to randomize enough of them to achieve that balance of measured and unmeasured confounders. So these are some of the ways in which non-experimental studies of treatment effects can be valuable. And I think you could probably come up with these based on the limitations that I just described. So I'll skip over that. I think it's important to understand why the result from a randomized trial may differ from that from a real-world evidence-based study for reasons other than bias in the real-world evidence study. And Sebastian highlighted some of the investigations that they undertook to try to understand these in the work that they're doing. So one reason may be that they actually are answering different questions. That is, one may be estimating the treatment effect in a treated population. The other may be estimating the effect in a total population. Another reason might be that the actual magnitude of the treatment effect differs. And if that is the case, you will get a different mix of those individuals, and the average treatment effect is going to be different. One is that the treatment itself may not be identical. So for instance, if you've been looking at a vaccine trial and you've administered 2 doses exactly 28 days apart, and you compare that to real world evidence where the 2 doses were administered up to 8 weeks apart, those are reasons that these effects may differ. And then there's also impacts of differences in adherence. And I described something here. And then not on this slide is that particularly in the case of a small randomized trial, there may not be sufficient numbers of individuals to actually achieve balance on confounders measured and unmeasured. And in the case that a real-world study were conducted, we would obviously statistically control for those differences and potentially arrive at a different result. So this is not to say that all real-world evidence is unbiased, but there are legitimate reasons to consider why the results may differ. So I hope takeaway number one, that I argue that yes, real-world evidence must be used to help answer many questions that cannot or will not be resolved by clinical trials. And I think it is unethical not to use the data from real-world experiences of patients to try to answer those questions. Now, we can't do that in a way that ignores some of the real potential sources of bias, so we have to do our best to do that well. So when it comes to using big data, we love this because it looks like it's going to be easy compared to conducting a trial. The data are already out there and the numbers are huge. And as Steven suggested, that we have these incredibly large sample sizes. And the problem with this is that as sample size increases, the random error component of our estimate decreases dramatically. But systematic error does not decrease with increasing sample size. So those sources of bias that may have crept in are not going to automatically go away as the sample size of our study goes up. So precision is, greater precision generally follows with greater sample size, or specifically with a larger number of outcomes, and that reduces the random errors. So in these two studies being compared on this forest plot, the top one had a sample size of over 300,000. The second one had a sample size of over 300. And you can see how much wider the confidence intervals are on the second one compared to the first. This also means that a, quote, highly statistically significant effect may not be clinically meaningful, and it's not an indication that the result is accurate. So precision is not the same as accuracy, and we can have a precise but inaccurate result that represents, for instance, a very tight cluster here. We can have an accurate but imprecise result that is centered on the truth. It is unbiased, but it will have wider confidence intervals. And then obviously what we're all hoping for is a precise and accurate result. So accuracy is the absence of systematic bias and can't be judged by the result itself. As we just reviewed, there are many reasons why our randomized trial may not serve as a gold standard that will let us decide whether or not our real-world study is giving us the truth. And so the way we need to judge the accuracy is actually not based on the result, but the process that arrived at that, including our understanding of the data, the use of an appropriate study design, and rigorous analytic methods. And as others have mentioned, most of the samples of the effect sizes that we will be looking at in real-world data may be in the range of 1.2 to 1.4. So if we don't appropriately address systematic bias, we can end up in a meaningfully different place in terms of that effect estimate that would really change our conclusions. So takeaway number 2 here is that precision is not the same as accuracy. And I would say that precision without accuracy is an illusion of certainty. So it would appear that you have very little uncertainty based on this very tight confidence interval, but if that does not reflect the uncertainty that's actually there due to potential systematic errors. So let's talk about how we limit those systematic errors, because these are the threats to accuracy that we really need to be tuned into. Accuracy is the result of a process and expertise. And I want to highlight how that works in the real-world evidence setting compared to randomized controlled trials. So there are really 3 primary categories of systematic errors for us to think about. information bias, confounding, and selection bias. And I'll touch on how we address each of those three. So in the same way that there are standards and processes for conducting randomized trials to ensure that the assumptions needed for causal inference can be made, there are analogous standards for conducting non-randomized study. And if we meet those standards, then causal inference is possible in non-randomized settings, I would argue. So in terms of combating information bias, on the left here, I show sort of what the processes are that are used in randomized studies. In non-randomized studies, we really are aware, obviously, that our data are not perfect, and we lean more heavily on hard outcomes, such as mortality. We have a deep understanding of how those data are generated and how the algorithms that we use to derive information from that perform, whether those perform differently in different patient populations or different treatment groups. And then we have to use appropriate analytic methods to account for those imperfections, not simply taking the data as presented as a given truth, or analyzing the complete data and dropping anyone who has partial missingness. So confounding is the sort of second of the three major threats. And in a randomized study, it is the randomization itself. If you have a large number of observations that achieves balance, and we evaluate that by looking at table 1. So we can sort of lay eyes on table 1 and say, yep, those look like they're pretty similar on all the things that we did measure. In a non-randomized setting, we do a lot of things with the study design phase to ensure that those individuals who we are analyzing are going to be as exchangeable, as comparable as possible at the start of our study. And that is, they have similar indications for treatment, they're at a similar disease stage, and that they aren't too frail to be eligible for treatment. And then, of course, we measure to the greatest extent possible those factors. We statistically adjust for them. And then finally, it's important to kick the tires, conduct sensitivity analyses to really understand the potential magnitude and direction of any uncontrolled confounding. So then the third major sort of category of bias that we have to worry about is selection bias. And by selection bias, I mean the fact that potentially not all individuals will be followed for the entirety of the follow-up period. And those who can be followed may be fundamentally different than those who disenroll or are not followed for the entire period. So in a randomized study, as I said before, we would ensure near perfect follow-up. We have a run-in period so that we eliminate individuals before randomization who might not be able to comply with all of the requirements of the study. We even restrict the population to those without comorbidity so that we don't risk losing them to sort of other complications that are related to their health. In the non-randomized setting, we have to plan very carefully, both in the design and the analysis phase, how we're going to handle patients who change or stop therapy and those who aren't followed for the full follow-up time or die before the end of it. And when I say plan carefully, I don't mean that we throw away everyone who can't be followed for the full time or who dies before the end of follow-up. There are specific approaches to handling those to ensure that we don't introduce bias by only selecting people who remain healthy enough to stay in our study. So the two problems that I see routinely crop into observational studies are crystal balls and time travel. These are sort of my terms for these two problems. And a crystal ball is when an investigator is using future information about a patient's treatment or outcomes to assign their status at an earlier point in time. So this is a crystal ball in the sense that if you had a patient in front of you and you were making a treatment decision for them, this is information that's not knowable at the time that that decision is made. The other problem that I see routinely is what I think of as a time travel problem, where we create a comparison of two groups of patients who can't possibly be compared with each other because one group would have to travel back in time and change their history of treatment in order to match that of the comparison group. So these are two problems I think that happen a lot and that account for a lot of the bias that we sometimes see. So this is sort of the framework that we can think of these both data and methods considerations in, and this is a figure that I adapted from a Duke Margolis white paper. And these fall under data considerations, that is, are the real world data of sufficient quality? And then methodologic considerations. So how do we design the study and analytically address it? And if those are handled appropriately, we, I believe, can develop fit for purpose real world evidence. So the third takeaway here is that developing robust fit for purpose RWE requires both a deep understanding of the data sources and the context in which they were generated, as well as appropriate application of tools and methods that are specific to this type of study. And both 1 and 2 here take time. So these are not turnkey, fast, push button study designs. We could also add peer review to the list of things that take time. So there are a lot of papers that are being posted on public archives that they haven't been through this process yet. And it does take time for the sort of the peer review process to take place. So my final slide, these are just sort of the high level conclusions from what I've already highlighted before. But I would argue that we have an ethical obligation to use real world evidence to understand both the benefits and the potential harms of treatments that are being used in the populations who are using them. And we do have the methods and specialized knowledge to do this work well. It doesn't mean that everybody who's publishing is doing it well, but I don't think that absolves us from trying.
Okay, Dr. Johnson-Funk, thank you very much. So we have 15 minutes for some questions. If you'd like to get your question in, put it into the Q&A text function or raise your hand. But I would just like to start with Dr. Schneeweiss. This question is obviously a precursor to what would be a much longer debate. But you saw Dr. Nissen's talk, and his skepticism of real-world evidence. I mean, what are the 2 or 3 takeaways from his talk that you think, that how would you respond to him in the kind of the 2 or 3 most important ways?
Yeah, so Dr. Nissen did a very good job in channeling the usual concerns that we hear in the community. So that's nothing new. What is important, I think, for this audience to understand when we talk about randomized controlled trials, it's not just about the random treatment allocation. It's equally about that most of the time we have primary data collection. It's about the data as well. While in real world evidence, often we work with secondary data, with data that were generated by the routine operation of the healthcare system. We're using that now for research, which comes with a whole host of issues. And there we talk about the data need to be fit for purpose. And Michelle touched on that quite nicely. We have learned a lot. We have come a long way from the HRT story. We know now what went wrong with HRT, with these analyses. The Framingham Heart study, by the way, got the finding right. They were spot on because they made a different design choice. They used the new user study design rather than this prevalent user design that was used by the Nurses Health Study and others. So we have learned our lessons. We still need to do a better job in educating the community of applied researchers. And I very much agree with Steven about the many studies that are of bad quality in real world evidence. They're muddying the water. And it's difficult for an audience like this audience here to distinguish between the good real-world evidence, the bad real-world evidence, the good real evidence that follows the guidance that Michelle laid out so beautifully, right? Versus somebody sees some data, does some analyses, and you get, colorful charts, you get numbers, but you don't get insights really, right? And in this, I'm so grateful that we have this session here in order to educate you and provide you resources where you can read up more.
Okay.
Do you mind, Chris, if I follow up on that?
No, go ahead.
I think one of the real challenges that Steven, your examples show is that in settings where we can't identify new users, we are really hamstrung by the fact that the effects of those exposures have already taken place, and we can't really temporarily separate the ongoing effect of, say, a dietary choice to use diet sodas on obesity over time. There is no time zero that we can accurately identify. And that is one of the sort of the core foundational study design principles of this field of research for looking at treatment effects, is that we need to be able to identify that period of time when someone is at a treatment decision and initiates treatment A or treatment B. And a lot of the nutritional epi studies, understandably, really struggle with that because it's not really an option unless you're going to do a full randomized study.
Yeah, I think both of you make very good points. And let me just comment. I made a case because I thought that it was important for the journalists to hear it. I recognize that there are people that do a much better job and people that don't. Here's the problem is, at least once a week, I get a call from a journalist and they got a press release from X university that said they did this study and they showed this finding, and the finding's just complete nonsense. And I often don't get called, which is even worse, because then I read in USA Today or, all kinds of high quality, newspapers, or watch it on television, and the people and the public believes it. And so I wanted the journalists to understand that the vast majority of observational research does not show causal high quality evidence. It's poor quality evidence. And we've got a small fraction of people like the two of you who understand all the things that can go wrong and work very hard to get it right. But that's not what the journalists are seeing. And they've got to understand the difference. Now, I will be skeptical of any real world evidence because that's my nature to do that. I also recognize that what you said about the fact that you can't study everything with a randomized controlled trial is absolutely right. And so if we're going to do any of this, we've got to do it well.
Okay, well, Dr. Nissen, let me ask you a follow-up question. What do you think, what do you think the motivation is now for, this move or this pressure to explore or amplify the use of real-world evidence? Is it to gain important information, or is it basically to save some money on the cost of doing randomized controlled trials?
I think it's mostly about money. I'm going to be very, very frank about this. Look, I think there is a better, I think there is, as I said in my final comments, what we really need to do here is those things that can be studied with a randomized controlled trial, we need to figure out how to do them less expensively. Simple case report forms, simple follow-up, easier, better, faster, cheaper. And if we can't do that, then we've got to do some real world evidence studies, and we need to look at all those sensitivity analyses that both of you talked about. We got to look at methodology. They've got to be very carefully peer reviewed, and we have to be skeptical if the effect size is modest. If the effect size is big, maybe we can believe it. And I'm talking about a twofold or 0.5 relative risk, not a 20% difference in relative risk.
Okay, so I have another question for you, then I'll go back to Dr. Johnson, Funk, and Schneeweiss. The HRT example is, is obviously very complicated, and there's some, interactions in the chat that the viewers should call up to get some more background. That is 20 years old. How has data science changed enough in the last 20 years that, legitimate problems that were done back then, because of the changes in data, render them, less concerning?
It's actually gotten worse, much worse. What's happened is the availability of electronic health records has made it possible for everybody who wants to do a poor quality study to do it. And they just pull up the electronic health record, they ask a bunch of questions, they analyze the data, and they publish it. And so the filters are largely gone. In the bad old days, you had to really work to get the real world evidence kind of studies done.
Mm-hmm.
Now it's too easy. And so the quality has actually fallen off.
Okay, let me go back to Dr. Schneeweiss with a different question. Your examination of 30 randomized control trials, the replication studies, you've done 20 of the 30 that have been released, is that right?
That's right.
Okay, and the remaining 10, are those the biggies, or is it just kind of a matter of chance about which ones have come out and which ones haven't?
It just continues. There was no specific reason why it was ordered the way it's ordered.
Okay, and for the journalist who's covering the issue of real world evidence, I mean, what are the questions that they should be asking if they see that there has been, a new study, based on real world data, or an FDA approval that is based on real world data, real world evidence? I mean, what are the questions that journalists should be asking so they properly understand how this is different than what a randomized controlled trial may have been?
Right. So the flip answer is, well, you've got to read a textbook of how to do these types of studies. Of course, that's not helpful. I appreciate that. What I see as a very useful thought paradigm, at least, is that you contemplate the randomized control trials that you would like to do but cannot do, I was not done, and then put these two things next to each other: what the real world evidence study looks like, and what the randomized trial would look like. We sometimes call this the target trial that you're trying to really do, but now we do this with secondary data in the absence of baseline randomization. And this will often raise questions about the validity of the study, validity of the design, validity of the underlying data sources. Could we really capture more subtle things about functional endpoints, the cognitive endpoints and things like that? Can they be captured with these data, right? In randomized trials, you go out there and the physician, the investigator has a specific list of items that should be asked from the patients. We don't have that when we work with secondary data. I want to respond also to Steve with regard to healthy skepticism about real world evidence. I totally appreciate it, and we need to do this. I just want to make sure that we also have a healthy skepticism about randomized controlled trials. There are enough of those that are not very well done, right? And if you think about the recent Alzheimer's medication, where you have 2 trials that were, I don't know, it looks like almost identically done, have 2 very different findings. What do we make out of that, right? So the world is not as simple as we would like it to be clear, right? Again, but this target trial paradigm, the logic of randomized trial is very appealing, very straightforward. Everyone understands. My mother understands a randomized control trial. She's not in the medical field. She's in the mid-80s. She's sharp, but still. So there is a lot of appeal to that. But the foundational thinking that Michelle pointed out very clearly, it's unethical if we have questions that we say we cannot answer the question because we don't do a randomized trial. And at the same time, all the data are lying there. Not doing anything here is not an option. We need to do it better than we often do today.
And I might add,
Sure.
Sebastian, that, as a journalist, I would find it incredibly daunting to try to read these papers based on real world evidence and make a judgment of whether or not there's sufficient sort of thought and quality. We have an entire PhD program to train students how to conduct these studies and to do it well. And the reality is you need to talk to an expert in the design and conduct of this particular type of research. And the training that, for instance, a biostatistician has who's primarily focused on randomized trials is completely different than the training of someone who's involved in the design and analysis of real world data. So it is a specialized set of knowledge and skills and design choices, methods, and you need to talk to somebody who has that training in order to evaluate sort of whether or not this study sort of lives up to those metrics.
And that's why I want the journalists to be skeptical when they see these kinds of data and make certain that they don't get seduced by low quality data into writing a headline that's going to mislead the public, because that's happened a lot.
Yes.
Okay. And with that moment of agreement, we need to draw this one to a close. I want to thank all three of you for your time and expertise. For the journalists on the call, we packed a lot of information in here, and the publications that some of the panelists were putting into the chat room, we will get those onto our resources list also. So if you want to look back at the HRT, the hormone replacement therapy debate, we'll have some resources for you on that. But Dr. Schneeweiss, Dr. Johnson-Funk, and Dr. Nissen, I want to thank you all very much.